or scheduling, or can I change this, can I change that? Again, I'm a guest lecturer. I usually lecture first and second year medical and dental students. And I teach mostly immunology, some microbiology. Now, every year they ask me to give several lectures in immunology, so what I try to do is try to compress as much as I can into 7 lectures. I mean, as you can imagine, giving immunology in 7 lectures, like giving biochemistry in 7 lectures. So, I mean, our student meds and dents, they usually get about, if you include the pathology of inflammation, they get about 30 hours of immunology. So what I try to do is I try to emphasize the highlight of an immune response, okay, and go from there. Now, so the immune system is basically a defense mechanism. Obviously, you must know this by now, you know, from a biology class. But it really is also a mechanism of recognition. And recognition is vital to your survival, okay? In everyday life, you have to be able to recognize things, okay? And sometimes it's components. So when you get out of here, you have to be able to recognize the door. And in order to do that, you have to be able to recognize the rectangle shape of the door and differentiate it and the rectangle shape of the screen so you don't bump into the screen, try to get out. Now, this is all self-evident to you, but remember that you have learned this, okay? You have learned this. If you ever seen one of those toddler games when you have little pegs of wood in different shape and little holes in a little flat thing, so you have, you know, a round hole and a round peg and a square hole and a square peg, and the little toddler is trying to fit a round peg in a square hole. Tuck, 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 tuck. Ah, this kid's stupid. Your side of the family, honey. Okay? Kid's not stupid. The kid's trying to learn. Okay? And believe it or not, your immune system has a mechanism we'll talk about when we talk about tolerance that learns. Okay? Now, the problem is that in learning, okay, there may be some misinterpretation. So if I have to tell you one single message that's very important, is the immune system doesn't know what's good or bad for you. The immune system can only tell what belongs and doesn't belong. And that concept of not belonging is biased, okay? And if that interpretation is wrong, then you have problems like autoimmunity or hypersensitivity, okay? So the immune recognition at the cellular level is also complex and biased. Okay. So I'm going to show you a picture. This happens to be my son, plug for my family, you know, nothing major relevant. Oh, now I hear comments. I hear somebody laughing or giggling. Why? You didn't react to the first picture, but you react to this one, okay? Your reaction is different. Now, let's say that I tell you that this dude right here is a Catholic bishop of Cleveland, or the junior senator from Oregon. Somebody laugh. Now, ain't nobody here that knows what a Catholic bishop of Cleveland looks like, okay? But you don't believe me, you laugh. Now, why do you laugh? The reason why you laugh is because you take this picture and you break it down into its components, okay? You look at it, you break it down to the jewelry, the tattoo this guy has. By the way, it is a guy. Okay? And then you have through some sort of screening system in your mind that is based on what you learn, okay? You have tools that look at this guy, your eyes in this case, and then you reassemble the picture in some sort of cognitive conclusion telling you, ain't no way this guy could be a senator or a Catholic bishop. Okay? Okay, second picture. Now again you laugh, but why do you laugh? I mean, this is a common character. Everybody knows this is a stormtrooper from Star Wars. There's nothing funny about a stormtrooper from Star Wars, except for what he's doing. Because now you analyze this picture in a context. Okay? And what you do is, again, you take the picture apart, okay, using certain receptors, in this case your eyes. In everyday life you may include your touch, your smell, your hearing, okay? And then you channel it through some sort of filtering system, okay, that somehow makes you laugh because this doesn't belong, it's not expected, okay? Now, logically, you accept this, you know, because everybody does that, right? Especially us guys with a newspaper in the morning, it's nice and cozy, but you don't expect this in a picture of a stormtrooper. So in your analysis of this picture, this doesn't belong in the expected thing and therefore creates laughter. Now, if you go back to the guy with all the tattoos, if you show that to a 1-year-old, the 1-year-old will probably just say, oh, far out, cool colors. That's it, okay? So the whole idea of the immune system is it's a process of recognition and a constant fight between recognizing and avoiding recognition. And there are many pathogens, okay, which I won't have the time to discuss. If you come to medical school, we'll discuss it at length, that are very clever in avoiding recognition by the immune system, okay? All right, so in all this concept, what I want to cover today, which will be very mercurial and strange to you, and hopefully will come to fruition in the next few lectures, is what are the recognition receptors that are involved in this sort of recognition, molecular recognition. In everyday life, it's your eyes, it's your ability to see, it's your ability to hear, to smell, to touch, to taste, and then you have a processing center in your brain that somehow concludes something from these receptors being activated. So the first set of receptors or molecules that we talk about in the immune system are antibodies, immunoglobulins. Now, once again, because there is such a variety of different backgrounds in this course, okay, some of you may find this very simplistic. On the other hand, some of you may find it complex, okay? I try to stay in the middle here. So antibodies, okay, play a major role in the immune system, okay, because they attach to foreign things that are called antigens. Now, they also, if you ever will continue in your clinical world, in the medical world, you will also find antibodies in many other aspects of both research and clinical entities. Okay, for example, diagnostic kits. Okay, as a physician or a dentist or a nurse or an NP, you will order diagnostic tests that are using antibodies to detect a particular entity, okay? Even in your everyday life, okay, especially in your age group. Have you ever sweated over a pregnancy test? You know, one of those, oh, God, please, 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 please. At that very moment, you may not be keenly aware that there's antibodies involved, but there are, okay? So you will encounter antibodies just about every aspect of your life. Now, major concept. Antibodies carry two major functions, very important ones. First one, obviously they bind to a specific antigen. The antigen is the foreign entity that you have decided that it doesn't belong. But secondly, they also activate other immune mechanisms. Once they have bound an antigen, okay, they turn mechanisms on that in turn act upon that antigen. Okay? They act like red flags on that antigen. They act like labels on that antigen. They make that antigen more visible to the phagocytic system, for example. Now, this is a basic structure of an antibody molecule. Okay? And again, I'm trying to be brief here because we have to cover a lot of molecules. So it's basically a molecule composed of 4 protein backbone chains, heavy chains, the yellow and whatever it is, the brown one here, and light chains. Okay? This is a basic subunit structure of all antibodies. Now, incidentally, this type of structure is also shared by a lot of other different molecules beyond antibodies that are involved in the immune system. The T-cell receptor looks like that. Some of the components of the major histocompatibility complex, which I will cover later in this lecture, kind of look in that shape. Okay? Some of the adhesion molecules that cells use to locomote through the blood vessels have similar shapes. Now, just to show you, these are not sticks in space, okay? This is a three-dimensional picture of the same thing. The yellow one here is a heavy chain. This thing here, what is it, mauve, whatever the hell that is, okay? And that's also the other heavy chain, and the red ones are the light chain. There is a variable region and a constant region, okay? This is where the antigen binds. So the constant region is a region that doesn't change much from immunoglobulin molecule to immunoglobulin molecule. On the other hand, the variable region, which is usually associated with the binding side of the antigen, as the name implies, varies from molecule to molecule. Now why? Well, because different molecules are created to bind different antigens, okay? And this is where the antibody diversity is created. We'll talk about this in a minute. So when you're making antibodies to an antigen, the variable region varies according to the antigen that has to bind. Okay, I talk about heavy chain and light chain, so I'm not going to talk about that too much. Now, the other thing about an antibody molecule is some of those molecules have what is called a hinge region, okay, which, as the name implies, act as a hinge or a flexible segment of the molecule. And what happens is that it allows variation in space between the two sides of the molecule that bind the antigen. So it allows flexibility. Molecularly, these are flexible segments of the heavy chain. So imagine an antibody molecule is like the eyes of a chameleon. You know, a chameleon sits on a tree, and one eye is looking for food, the other eye is keeping an eye on his girlfriend, and they're, you know, kind of like independent. The same thing happens with an antibody molecule. So it allows some flexibility between the binding sites to adapt to different antigenic situations. Now, in humans, you have 5 classes of immunoglobulins. Okay? Name Ig for immunoglobulin, IgG, IgM, IgA, IgE, and IgD. The main differences among these folks are, 1, there are structural differences, there are similarities in the basic unit, but there are structural differences, and more importantly, they have different jobs. Okay, and I will discuss each isotype or class. So IgG is the most common immunoglobulin in peripheral circulation in a normal individual, okay? It's about 150,000 Daltons of molecular weight. No, I'm not going to ask you to remember 150,000. I'll just give you a reference point to the differences in sizes among the different classes. Okay? And what IgG does a lot of different things. For example, it can neutralize a toxin. Okay, by binding to the toxin, changing its shape, or binding to the active size of the toxin, and therefore render it untoxic. Is there a word like that? You know what I mean. It can opsonize. Opsonization is basically the binding of opsonin, such as antibodies and certain complement components. We'll talk about complement in a different lecture. So that the antigen is more visible to the phagocytic system. Okay? Opsonization, think of it like the ketchup of the immune system, where the item to be recognized is now more recognizable because it is covered by things like IgG, okay, which in turn can activate things like phagocytic cells. It can agglutinate, it can clump up things, and once things are clumped up, they're more visible to the phagocytic system, and it can activate the classical complement pathway. Once again, we'll talk about complement in a different lecture. 2 immunoglobulins classes can activate the complement pathways, IgG and IgM. The other 3 do not. Okay, write this down. Now, the second immunoglobulin is IgM. As you can see, IgM is much larger than IgG because in general, it occurs as a pentamer of 5 different basic units of immunoglobulin linked by the sulfide bonds, okay? Now, evolutionarily, is the most conserved immunoglobulin. It's a primordial immunoglobulin. Okay, for example, it's the first immunoglobulin to appear in an immune response the first time you see an antigen. So in a primary antibody response meaning the first time you encounter an antigen, the first wave of antibodies to that antigen are usually of IgM class. It is also the first immunoglobulin produced by neonates. Okay, as they develop their own immune system, okay, neonates usually receive some maternal immunity via transfer of placental circulation, so they have maternal circulating antibody, and also during breastfeeding they get some antibodies from a mom that way. But when they develop their own immune system, usually the first immunoglobulin to appear is IgM. Okay, IgM also can neutralize toxins, can opsonize, can agglutinate, and can activate the classical complement pathway. Now, in addition, okay, this I'm talking about secreted IgM. In addition, IgM as a single molecule can also occur on the surface of B cells. We'll talk about activation of B cells that act as a receptor for the B cell to recognize a particular antigen. Okay, IgM and IgD do that. And here's a picture of IgM in the pentameric form. It kind of looks like a ninja star, heavy chains, light chains. These are the classical immunoglobulin subunit, and this is in pentameric form. And then you got IgA. IgA is the predominant class in extravascular secretion. What are extravascular secretions? Mucus, saliva, tears, breast milk, okay? All those contain IgA in the form of secretory IgA, okay? So it's very active in your gut, into your genitourinary tract, into your respiratory tract, in your lungs, okay? In the mucus, in the respiratory pathway, okay? In your mouth, in your saliva, you have a lot of secretory IgA. Okay, and this is what it does. Once again, in extravascular secretion is the major antibody class that is active against antigens. Also, in colostrum, it transfers maternal immunity to the neonate postpartum. Okay, when a mom breastfeeds a baby, of course, she gives him or her nutrition, of course. Okay, but in addition, she also gives the baby various different immune components, various different immune cells, including secretory IgA. Okay, and because many pathogens enter the system via the mucosa, okay, it kind of gives the baby a coating of antibodies, okay, to give the baby a chance while it develops its own immune system. Okay, it can neutralize toxins, it may opsonize, but it does not activate complement. Only IgG and IgM do that. Okay, and here's a picture of secretory IgA. Okay, two subunits right here. Okay, with a J chain and a secretory component. Now the next one is IgE. IgE was designed by Mother Nature to fight parasites, okay, worms, things that burrow into the digestive system. But in reality, nowadays, it's really the one that is involved in allergic reactions, okay? So if you have hay fever, if you have asthma, or God forbid, if you get into systemic anaphylaxis, IgE is the main culprit. Okay, this is an example. We'll have a whole lecture on hypersensitivity, where the immune system reacts to something that perceives it as a threat, is not really a threat, but in doing so, it causes more harm to you than the threat itself. Okay, the major function is to arm mast cells and basophils. These are cells that are involved in allergic reaction, to release a whole bunch of different chemicals, such as vasoactive amines, histamines, leukotrienes, that in turn cause the allergic reaction that you may feel if you have an asthma attack, or if you have a hay fever, or if you have atopic dermatitis, things like that. Okay, and this is about the size of IgD. IgD is very low in concentration, is difficult to study. Its major role, like IgM, is to act as an antigen receptor on B cells. Once again, B cells recognize an antigen by having surface of immunoglobulin, immunoglobulins that instead of being secreted, okay, they are anchored to the membrane and recognize an antigen. The classes of these immunoglobulins can be different ones, but the major ones are IgD and IgM. And here is the cast of characters right here. Okay? So this is one set of eyes that can look at something, okay, and allow recognition of a particular antigen and response to that antigen. Okay, this is dead. This will move. Okay, once again, there's also membrane immunoglobulins, and once again, those act as antigen receptors on B cells to recognize an antigen. Okay, I'm going to skip this. Okay, what they do, they are initial in antigen recognition, so a B cell will recognize an antigen via these little radars that they have on their surface. And once that has been achieved, then in turn, membrane immunoglobulins also activate the B cell, tell the B cell there's an antigen, act upon it. Okay? Those are the major roles of membrane immunoglobulins. Okay, and here's just a picture of a membrane immunoglobulin. The only difference between a secreted IgM and a membrane IgM is a stretch of amino acid that happen to be hydrophobic, and therefore they're very happy in a plasma membrane. It kind of anchors the molecule to the membrane of the cell. Okay, and here is another picture of a membrane immunoglobulins. Membrane immunoglobulins are coupled to other molecules. Once this is activated, it transmits signals that in turn activate the cell. Now, the other set of eyes are T cell receptors. Okay? T cells and B cells are critical cells of the immune system. We'll talk about that in a different lecture. Like B cells recognize antigen via their surface immunoglobulin, T cells also have surface receptors called, cleverly, T cell receptors, which recognize antigen. Now, when we talk about antigen processing, T cells recognize antigen in a much different way than B cells. Okay? B cells usually recognize antigens in the way they appear on the surface when they first come in. T cells, on the other hand, recognize antigen after they have been chemically altered and presented by an antigen-presenting cell. And we'll cover that in a different lecture. Right now, just bear with me with this molecule. Hopefully the story will come together in the next few lectures. Okay? So just like B cells, T cells also recognize antigens, once again, via their T cell receptor. Okay? Now, the receptor is made of heterodimers, meaning there's two of them, dimer, and they're different. And this is what they look like, okay? They have a variable region, they have a constant region, they have a transmembrane region, okay? This is the area where the antigen binds. This is the membrane of the cell, okay? And these are also coupled to other components of the cell that when this T cell receptor is activated by an antigen, also get activated and transmit signal to the cell to do something, okay? Once again, if you have a little radar on the surface of a cell, you have to have wiring that connects that radar to the inner mechanism of the cell so that the cell can do something to respond to the signal that has been detected by the radar. Okay? So we have T cell receptors, and we have B cell receptors, and we have surface immunoglobulins, secreted immunoglobulins, okay? Now, all these are chemicals that are able to detect a particular antigen, okay? Now, this comes to a very complicated issue, okay? Because what happens is that you have a lot of different antigens that you are likely to encounter in life. They're all different. So how can you have so many different antibodies, so many different T-cell receptors, okay, that are supposed to recognize the unknown? And this is a very important point. So I'm going to wake you up for a minute, okay? This is a very important point because this leads us to autoimmunity, to hypersensitivity, okay, to self-reactivity. So the way that the body, the immune system works in encountering the unknown is that it counts on a concept that nature is made of repeating patterns, okay? So let me give you an analogy. Think of music. Now there's all kinds of different music, right? You have Vivaldi, you have Beethoven, you have Shaggy, you have Lady Gaga, right? Okay, you have Psy, Pavarotti, Oh Sole Mio, and then you have, Shaggy, they call me Mr. Bombastic, right? Right? And Lady Gaga, Ra Ra, Oh Mama, Gaga, Oh La La. They don't sound nothing like it, right? Okay, and then you have Psy, okay? I'm a Gamma style, right? Well, you laugh, but what do they have in common? Think about it. They sound nothing like it. They're all algorithms of the basic 7 notes. And no matter how much you twist the music, you are bound by simply having different patterns of basic units. Okay? So it's to have basic limited number of units and arranging a combination that are all different. Now think of what we know in nature. Okay, we don't have an infinite number of things in nature. They are limited by the 100, whatever they are next Tuesday, elements of the periodic table. Right? And even how they get together, they're limited. So for instance, you can take sodium and chlorine and make it sodium chloride, but you cannot take sodium and neon and make sodium neonate. There's no such thing. Okay? So the way that the immune system works is that it counts on the fact that no matter what I encounter, it may have a repeating pattern somewhere. Okay? So think, for example, of an antibody and antigen as a lock and key interaction. Okay? You may think that you can make an infinite number of locks out there, or an infinite number of keys, but actually you can't. Why? Because you're limited by how many grooves you can put in a key, okay? How many teeth you can put in a key. So the way the immune system works, believe it or not, instead of preparing for something to encounter, okay, what it does is it sits at the key-making machine thinking, yeah, there's a lot of locks out there, but not infinite, okay? And keep making random keys. So there's some dude sitting in a key machine, making random key patterns. And as locks approach you, okay, as antigen you encounter, you try the key. And if a key doesn't work, you throw it out. But if the key works, now instead of throwing it out, you save it as a memory cell, and you keep it in a pocket. And that cell replicates and makes a lot of keys of the same specificity. So now if that lock encounters you again, now you have many keys for it. You don't have to shuffle through a boatload of keys. You have the key ready. And this is how the immune system works. It makes randomly, constantly making keys that I keep throwing away. So now the issue is, how can you make so many keys? And the way that it works is the following. And by the way, this is a critical point because, as we will see, sometimes you don't have the right key for the right lock. So the way that it works is, well, maybe there is a gene in your body that accounts for every single possible immunoglobulin molecules you're going to make. Well, that's highly unlikely, because if you did that to encode an entire immunoglobulin molecule, okay, with one gene, half of the genome would be occupied by immunoglobulin genes. Of course, your genome encounters other things. So people then started thinking, okay, maybe there's a low number of genes, And because you're making random specificity, random keys, maybe somatic mutation, which is an event that occurs on a random basis, changes these particular sequences to make random specificities. Well, the problem is that you would have to have, again, an enormous amount of somatic mutations in order to do that. So then people looked at an immunoglobulin molecule, especially its single chain, and what does the chain look like of an immunoglobulin? It has a constant region and a variable region. And where do you have to create diversity? In the variable region. Why? Because that's where the antigen binds. So people thought, oh, wait a minute, this kind of looks like a screwdriver. Okay, a screwdriver has a tip and a handle. And the handle doesn't change from screw to screw. The purpose of the handle is to act as leverage so you can turn the screw. What changes for a screw is the tip. So you have a flat head, you have a Phillips head, you have a torque, you have a hex, whatever screws there are. So somebody thought, well, maybe I don't have to have 100 different screwdrivers. Maybe I can have a couple of handles, and I can change the tip according to the screw. And indeed, that's what modern screwdrivers are like. Well, the same thing happens to an immunoglobulin molecule, where you have a constant region, which is the screwdriver handle, and a variable region, which is the tip that is adaptable to a different screw or a different antigen. Okay? So people realized that in order to create antibody diversity and T-cell receptor diversity, they had to go to rearranging the screwdriver tip, or the variable region of the immunoglobulin chain. Okay? And this is done in the following sequence. This is basic biochemistry, okay, where you have a germline DNA, your genomic DNA, that eventually gets, into the B-cell DNA, into primary transcript, into the messenger RNA, and this, of course, gets translated into the protein product. Okay, so here is a single immunoglobulin chain. This is the heavy chain, this is light chain. This is the constant region, this is the constant region, this is the variable region, this is the variable region. Here is the screwdriver handle. You don't have to change that for different screws. And here is the screwdriver tip. This is where you need the variability. This is where you have to create random keys in order to adapt to different antigens. So in order to create such enormous diversity, the first one is to have multiple germline variable region genes. Genes that encode for the variable region and have lots of them, okay? And attach them to a limited number of constant region genes. But that's not enough to create a very large repertoire. So then, what you do is you have other segments of DNA that are attached to the variable regional genes to create diversity. And one of them is the J segment. Okay? A J segment is an additional short segment of DNA that is attached to these variable region genes. And I have a picture of it. Okay? So the way that the immune system generates diversity, okay, is by having a large but limited number of components and arranging them randomly in different combinations. Okay? Think of a lock that has a numerical combination. Okay? If that combination of that lock has 2 digits, the possible combinations are 100, from 00 to 99. Now, just by adding a third digit to that combination, now the combination possibility goes to 1,000, from 000 to 999. If you get a fourth digit, now it goes to 10,000 possible combinations. So just by having a limited number of digits, you create a large number of combinations. So in the rearrangement of the variable region of the immunoglobulin molecule, here is a variable region genes. You have more than one. Here are imaginary 2. And then you have a bunch of J segments, okay? And just by mixing and matching randomly these, you create different combinations. So for example, in this combination, you have V2 and one of these J's to create this. But you could have this and this, or this and this, or this and this, randomly. So that increases your number of combinations at the variable region of an immunoglobulin molecule and also of the T-cell receptor. Okay? Now, in the heavy chains, there's yet another piece of DNA called the diversity, and you have a bunch of those. So now you add a third digit to the whole story. So here is now a variable region, okay? This is done. And here are the J segments, and here are the D segments. So once again, you can pick and choose, and of course there's a lot more than 3 or 4 of these. You can pick and choose whatever you match, this and this and this, or this and this and that, or this and this and that. And according to what you randomly pick, your final product is a large number or randomly chosen combination of variable regions, both at the end of the immunoglobulins and also of T-cell receptors. So different V, J, and D recombinations, by having a limited number of Ds, a limited number of Js, a little number of variables, and mix and match them in a random pattern, you create a large repertoire of final combinations. But that's not all. Okay, so here is heavy chain, here is light chain, and once again, don't let the picture scare you. Just depending on what you pick on these guys, you can get different combinations. Now, there's more ways to multiply. Come on, guys, wake up, geez. All right. Now, because this is a random pattern, okay, sometimes mistakes, she just got it. Sometimes mistakes, okay, can occur, especially when you're splicing DNA. Okay? So these inaccuracies in splicing are just as random as any other combination. Okay? So when you're making a key and you're making a key randomly, okay, you're sitting, you're going, oh, pizza's here. Okay? And then you get another key and somebody calls you, hey, Ernie, yeah, what? Oh, crap. Well, that's just as random, even though you made a mistake, as the one you just made. So you keep that too. Okay? And these joining inaccuracies sometimes can create slight difference at the protein level. Okay? Depending on how you attach these dudes to these dudes, okay, you can get different combinations just by splicing over by mistake, okay, and the result, as you can see, are different amino acids. And once again, these are randomly created splicing error, and because they're random, they're just as random as the ones that have no errors. So the immune system keeps them, again, as randomly made keys. Okay? Of course, somatic mutation does occur, not in enormous numbers to create entire diversity, but it does occur, okay? And that also creates random specificity, which the immune system works. And finally, all these are what? Are in a single chain, but immunoglobulins are made of at least 4 chains, 2 heavy and 2 light chains. So by also mixing and matching different light and heavy chains, you increase your number of possible random specificities. Okay? So here is what I just told you about the heavy chain with all the rearrangements. Here the light chain with all the rearrangements. And then you can take any one of these and any one of these and arrange them once again randomly, okay? So all these random patterns multiply the huge number of specificities that you may need to encounter what you perceive as the unknown, okay? Now, we will see what is relevant in this is because sometimes that goes against you. We'll talk about that when we talk about hypersensitivity and autoimmunity. Okay, so this is basically the summary of antibody diversity and also of T-cell receptor diversity. Now, those are the eyes, those are the receptors, but how do you make the decision? Okay, what is the filter that allows you to come to some conclusion to say, yes, I see the guy with all the tattoos and the jewelry, and I process this information, and I come up to the conclusion that could not be a senator or a bishop? Well, you have certain molecular things in your system that are involved in recognition and response to antigens after they have been detected by immunoglobulin. And these are the components of the major histocompatibility complexes. Once again, please bear with me, because all these will hopefully come to term in a mechanism when we talk about antigen processing and presentation. So basically, right now, I know how you feel, okay? Yeah, I know this is so 80s, but... Okay, the major histocompatibility complex is basically a bunch of genes. Okay, they encode for certain products. And what the products are divided into 3 classes, including structure and function differences, cleverly labeled 1, 2, and 3. Okay, and the one that we will discuss in antigen presentation, excuse me, are class 1 and 2. Okay, those are the ones that are involved, if you wish, in the somewhat decision of what to react against. Okay, so class 1 are those, please remember this, put an asterisk next to this. Class 1 are those that are involved in the processing and recognition of endogenous antigen. What is an endogenous antigen? It's an antigen that comes from within. For instance, alter self, or a viral product that has infected a cell and now makes the cell infected and dangerous, okay? Or a screwed up protein that the cell has made and has to be destroyed. Those come from within. Now, on the other hand, an exogenous antigen is an antigen that comes from an outside source. For example, a bacterium or a microorganism that enters somewhere and gets into your bloodstream, okay? And those are class 2. Once again, we'll talk about this when we talk about antigen processing and presentation. Okay? So MHC class 1 are basically composed of a single alpha chain attached to another component called beta-2 microglobulin, which is also involved in signal transmission to the cell. And this is found on members of all nucleated cells. Remember this. All nucleated cells have the potential of being infected or being altered, especially by viruses, and therefore all have to be potentially recognized as infected cells and be eliminated. So all nucleated cells express class 1. Okay, and this is just a picture of an alpha chain, okay, with a peptide binding clef. This is where the process antigen, we'll talk about this in a different lecture, binds. And here's beta-2 microglobulin. Now, class 2 is made of 2 polypeptide chains, okay, and it's found in all cells that are involved in antigen presentation. So only certain cells express class 2. Those cells express class 1 because they can be infected, okay, but class 2 is only expressed on certain immune system cells. Okay, and here is a picture of class 2 molecules. Here's a peptide binding clef. Once again, we'll talk about this. We'll talk about antigen presentation, 2 different polypeptide chains. Okay, now this is a very important point. I want you to remember this. Okay, put an asterisk next to this. You will constantly make random specificity of an immunoglobulin variable region and also of T-cell receptor variable regions. Okay, so you're constantly changing your repertoire. You're keeping the ones that do recognize a lot and keep them as memory cell. You throw the other ones away. On the other hand, the components of the MHC are fixed, meaning your repertoire of MHC is what you got from mom and dad. Okay, you're not making new ones all the time. You're limited. It's extremely polymorphic, so you have a lot of different alleles at each locus, okay? So you have a very large repertoire of MHC molecule that can react to a lot of different process antigens. Once again, we'll talk about that in the next lecture. But it's fixed, it's limited. You're not making new ones. And the significance of that is that you may encounter, because these molecules are involved in screening, okay, in conceptually understanding an antigen, if you wish, Okay? Even though you have a large repertoire, you may have a situation, because Mother Nature decided that way, that you cannot have a particular specificity for a particular antigen at a particular point in time, and therefore you cannot react to that antigen. Okay? And once again, we'll talk about that when we talk about antigen precision. So it's a very important point, because some people, for instance, believe that certain genetic populations may be more susceptible to certain diseases because they lack a particular specificity to particular antigen, to a particular pathogen. Okay? A lot of autoimmune diseases are linked to MHC. We'll see that when we talk about autoimmunity. Okay? All right. So I know you feel about this. Again, bear with me, because some of these concepts hopefully will gather strength when we talk about actual mechanism. Okay? Any question? If not, see you on Friday.